Hello, everyone, and welcome. It's my great pleasure to introduce our speaker today, Dr. Jason Hartford. Dr. Hartford is an incoming assistant professor in the Department of Computer Science at the University of Waterloo, and he will also be a Vector affiliate member starting July 2024. He's currently a staff research scientist at Valence Labs at Recursion Pharmaceutical. Previously, he was a postdoc with Yoshua Benju at Mila, where he worked on causal representational learning. Before joining Mila, he completed his master's and PhD at UBC, working with Professor Kevin Latent Brown, where he worked on deep learning-based estimators of causal effects and enforcing symmetries in neural networks. During his PhD, he had internships at Microsoft Research New England at Redmond, where he worked on deep learning approaches for causal inference. Please join me in a virtual round of applause to welcome Dr. Hartford.
Thank you so much for having me.
The floor is yours.
I'm really excited. Thank you. Thanks, Mark. I'm really excited to be here, and it's nice to be giving a talk at Vector, given that I'm going to be there. I'm going to be joining you guys today. I'm going to be talking about what I think is kind of the future of causal inference. But before we get there, we're going to kind of start with a kind of history of how we got here. And I'm going to be I will give you to sort of set the stage. Let's look at causal inference for the last 300 years in one slide, which is a massively overambitious thing to do, but let's try and do it. So for the last 300 years, if the world was too complicated to understand analytically, so if we're not a physicist who can sort of reason about how the world works based on a set of equations, the way we understood the world was by experimentation, right? So my slides are not working. This is good. Oh, there we go. Okay. So, in the sort of this early 1700s, people like James Lind were showing that citrus was important for preventing scurvy in sailors by running experiments and showing that it was really citrus that was the thing that was preventing scurvy and not just some acids. John Snow, who's well known as regarded as like the founder of modern epidemiology, showed that cholera was caused by dirty water by removing a pump handle in Broad Street in Soho in London. And it's kind of fun. You can actually go and visit and still see that pump today. But the way he was able to show that was by intervening in the world and manipulating the world and running an experiment and then seeing the outcome of that experiment. The sort of role of experimentation was formalized in the early 1900s through the work mostly of Ronald Fisher in his book, The Design of Experiments. And really, this was kind of the way that the causal inference was done. You ran an experiment, and that's how you found out how the world worked if you couldn't reason about it analytically. And this is an effective tool. It's still a very effective tool in our sort of toolkit of the way we understand the world. But the limitations of this purely experimental based approach were shown up in the 1950s with the debate around smoking and cancer. Right. So in the 1950s, it was well understood that there was a strong association between smoking and cancer. Cancer patients, lung cancer patients showing up in hospitals tended to be smokers. Smokers often ended up with lung cancer. But people like Fisher were vehemently opposed to applying a causal interpretation to this association, right? He would argue, look, he was also a smoker himself, but he would say, look, you can't rule out the possibility that there's some gene that makes me both more likely to get addicted to cigarettes and more likely to get lung cancer, right? And in a sense, he's right? Like that is true. And in fact, very recently, there are actually genetic associations between whether it actually makes you less likely to get cancer. We can go down that road. But broadly, this is something we have to be able to respond to, that kind of a critique. And so this raises the need for being able to draw causal inferences for observational data. Now, why do we need causal inference from observational data in this setting? Well, if you think about it, this is not something that you could ethically or practically test with an experiment, right? We're certainly not going to run a randomized controlled trial where I get half of teenagers to smoke and half of teenagers not to smoke. and see what happens, see how many people die. That just seems like something that should never pass an ethics board, even if we wanted it to. And then practically speaking, these effects take a really long time to show up, right? Cigarettes kill you slowly, and so you would need to run this study for a really long time in order to discover this, and we need to know these answers now in order to make policy changes to prevent further people dying from cigarettes, right? So this need spurred a lot of research in this area, and why are my slides not working? Sorry. Keynote is not operating for me. So in the 1970s through to today, there's been a massive growth in methods that allow us to do causal inference from observational data. So people like Yuda Pearl from computer science, Don Rubin from statistics, Jamie Robbins from epidemiology, Guido Inmans, Josh Angrist from econometrics. It's this sort of cross-disciplinary effort, have built out this toolkit of methods that we can draw causal inferences from observational data. And so now we sort of have all these different methods. And we can see the effects of these tools kind of nicely in economics, because at least from a computer science perspective, they have this like really unusual publication strategy where everyone wants to publish in the same journals, right? So regardless of what subdiscipline in economics you're in, you really want to publish in the same journals. And this lets us get a kind of imperfect estimate of the impact of causal inference just by looking at the change in the methodologies that are used in these papers that are published in these popular journals, right? And so that's what Bryce and Montesinos Yufa did in 2019. They basically made these word clouds of the titles from the 500 most cited journals from AER, QJE, and the Journal of Political Economy, which are 3 of those top 5 journals. And you can see that, from the 1950s to the 1980s, economics was a theory-driven field, right? So if you wanted to make a claim in economics, you would derive a theoretical model based on assumptions about human behavior, and you would sort of make your claim based on that. And then from the 1990s through to today, you see the rise of so-called evidence. And what economists mean by evidence in this context is causal inference, primarily from observational data, and then in the 1990s and 2000s, and then in 2010, experiments also became popular, right? And this was celebrated in 2021 with the Nobel Prize that went to David Card, who's from Guelph, Ontario, so just outside Toronto, and Josh Angrist and Guido Inmans. And so the point I want you to take from the slide is that these methods that, like, by giving a field like economics, or just more generally the social sciences, new sets of tools, you can completely transform how science is done in these fields, right? So developing new tools can change the way science is done. And this is in a field like economics is a pretty, they're a slow-moving, very careful field. They don't change direction very easily, and yet we can still see this sort of dramatic shift in the way they study the world. Which then raises the natural question of, well, we've done this with causal inference from observational data. What is the next frontier for a causal revolution, right? Where can we sort of build the next set of tools that changes how a field conducts its research? And I'm going to make the case today that biology is a really nice place that is sort of ripe for the influence of causal inference. Like economics, biology involves reasoning about the equilibria of cycles of complicated interacting systems. So you have these like really complicated systems that are very difficult to reason about analytically. What's nice is increasingly we can observe these biological systems in ever more detail, right? So the fun thing about being at Recursion is you get these fun videos, but this video is of the robotics platform that they use internally for capturing images of cells under a microscope, so cellular microscopy. And they can do this at scale, right? They can do massive numbers of experimentation in a highly automated fashion. And we have the ability to intervene. So we have the ability to experiment very easily in a way that is actually not true of the social sciences and economics. But the data modalities require new approaches to causal inference, right? The sort of data modalities bring new challenges that make the standard tools from the causal inference toolkit just not applicable. And so that's what we're going to be talking about today. And so I'm going to be focusing on two kind of distinct but important challenges that arise out of high-dimensional problems. The first is so-called high-dimensional treatment effects. And this is where you just have a lot of things you want to make causal inferences about, right? So you have a very high-dimensional system. I'll give you an example in a moment. And there's just, so there's just you have a lot of causal queries, and you have relatively few experiments that you can do. And so high dimensionality is just kind of intrinsic in the data. And then the second is high dimensionality that comes from the fact that we often collect unstructured data rather than structured data. So in those robotics platforms that we saw before, we're taking photographs of cells under a microscope, so we end up with pixel level data rather than structured data that you might get in a data frame that's sort of nicer to work with. And the question there is a representation learning question, which is like, how do we take that unstructured data and turn it into something more structured so we can do causal inference on top of it? Okay. All right. So this first part of the talk is based on work with Elizabeth Eiler and Nikki Kilbertus at Helmholtz Munich. And it's a paper that we wrote at ICML this year. And the question we were sort of motivated by this. So by these sorts of news articles you read. So imagine you sort of pick up the New Scientist and it says, and you sort of read an article like this that says, could your gut bacteria influence how intelligent you are? Now, we're all pragmatic scientists. If there's some gut bacteria that can make us smarter and let us get ahead, I'm sure we'd love to know about it. If only to just help us do our work better. But how would we analyze? How would we sort of study this rigorously? So this article sort of claims that there are a set of bacteria that give us better scores on verbal and mathematical tests. But if we want to go and test this, do sort of follow-up studies and test this, there's some problems that arise. So the main problem is the gut microbiome itself. It houses hundreds of thousands or millions of different kinds of bacteria, and we have sort of some health outcome of interest. So we have this very large set of potential causes for this health outcome of interest. So all of these different bacteria could potentially have an influence on this outcome of interest, and this relationship is clearly confounded, right? The body's mechanisms, environmental factors, and so forth are going to influence both. So, for example, our diet and the amount we sleep are likely to influence certainly our measured intelligence. If we're tired or if we've eaten poorly, we're probably going to perform worse on sort of standardized tests, and that's obviously going to influence the populations of gut bacteria as well. So it's very easy to think of lots of things that could potentially confound the relationship between these two variables, or this multitude of variables and this outcome variable. And the second problem is we can't sort of experiment on individual bacteria directly, right? You don't have the ability to sort of surgically go in and like kill one particular kind of bacteria in your stomach and leave everything else unchanged. That's just not an experiment that we can do practically. And so all we can do is we can kick these bacteria's populations around with different choices of antibiotic, right? So different antibiotics are going to differentially affect different subpopulations of bacteria. And by taking different antibiotics, we can change those populations of bacteria. And we want to see whether we can use that in some way to infer the causal effect of these bacteria on some outcome. Okay, so we have this large number of bacteria, we can run some experiments, we can measure the outcome. And we can obviously measure the populations of bacteria as well. That's set up kind of clear. I realize I'm kind of talking to blank screens, but hopefully that's clear. Please feel free to stop me at any time if this is not clear. The sort of uncomfortable thing about Zoom talks is I'm never really clear. I can't sort of look at you and see whether you're following. Okay, now if you've got a causal inference background like me, you'll spot that this setup that I've drawn here looks like what we call an instrumental variable setup, right? And in this case, the instruments are these antibiotics. So these antibiotics are instrumental variables kind of by definition. So what is an instrumental variable? It's some variable that only affects our outcome of interest, which in this case is intelligence in the sort of New Scientist article case, via its effect on the treatment variables, which in this case are the different bacteria, right? And to be an instrumental variable, it has to satisfy three assumptions. The first is the so-called relevance assumption. And that just says these arrows over here are non-trivial, right? So the antibiotics do actually push around the effects of bacteria in some way. So we're not just taking such a small dosage of antibiotics that it doesn't affect our bacteria at all. The second is so-called unconfounded instrument assumption. And this is satisfied trivially in this case. In more generally, if you're sort of using instruments in the wild, you have to worry about whether there's some confounding factor that led you to take the antibiotic and create an association with the body's mechanisms. But if we're running experiments, then this is going to be sort of satisfied trivially because we're randomizing over antibiotics. And then the final one that's always the hardest to satisfy is the so-called exclusion assumption, which we have to assume away the possibility that the antibiotics affect intelligence via some other pathway that is not mediated by these bacteria, right? And that is always the source of the biggest amount of debate. For the purpose of this talk, we'll just accept it, but it's the one that you should be most careful about when you're actually doing this type of thing in practice. Is there some other pathway that connects the instruments with the outcome of interest, because that'll bias your results. All right. Now, this is a setting that just hasn't been studied before, and we'll get to what it is precisely. So we're just going to focus on linear ID in this case, although I have done a lot of work on the nonlinear setting as well. And so in the linear case, how do instrumental variables work? Well, we can see this in a sort of linear 1D case first, because it's very easy to see there. And in linear 1D worlds, estimating the causal effect of the treatment variable X on the response variable Y corresponds to estimating this coefficient of a linear regression. So this beta coefficient of a linear regression, right? And so we want to estimate this beta coefficient of a linear regression. Normally you would just run a regression, regress X on Y. This is sort of, import your famous linear regression from the favorite linear regression package, scikit-learn's OLS, and regress these two things. You'd be fine if this wasn't confounded, but here we're going to assume that these two variables are correlated by some confounding factor. So this was the body's mechanisms that correlated these two things. And so a linear regression is going to be biased. So if I just directly regress these two things, it's going to be biased. So here's an example of this. So if I have no correlation between these two variables, we fit this is the sort of blue line is the truth, and the orange line is the fit we get. But if I turn up the correlation and to make them highly correlated, I can even flip the sign of the effect. So I can flip the sign from being a negative relationship to being a positive relationship by introducing a strong enough correlation between these error terms. Okay. So that's a problem of causal inference. That's a problem that we have to solve. And we have access to this instrumental variable, which we haven't used at this point. And this is the thing we're going to use. So how do we use that? Well, one thing we can notice is we can compute if we regress X on Z, so we get this, we compute the expected value of X given Z, and that's just going to be equal to alpha Z plus some bias term, plus some constant, okay? And similarly, we can regress Y on Z, right? And that's going to, and if we run that regression, we'll get alpha beta times Z plus some constant, right? And this is just by our assumptions, right? So you can just by plugging in linearity of expectation in our assumptions, you can get that, right? And so the simplest possible instrumental variable estimator you can use is the so-called world estimator that's been used since the 20s. And that just says, regress run these two regressions, first XZ and YZ, and then divide the two coefficients, right? And then you get sort of from the YZ relationship, you get alpha beta, and XZ relationship, you get alpha, and so dividing them will give you an unbiased estimate of beta, right? That only works in the scalar case because you're working with scalar parameters, but you can generalize this idea using a method known as two-stage least squares by noticing kind of the following relationship, right? So we can notice that in the data generating process, based on our assumptions, we can write the outcome variable y as being just a function of z rather than of x, right? So I can replace x over here with z. And the problem that arises is the confounding that happens between this epsilon x, this noise term over here, and this epsilon y, this noise term over here. So that correlation is what's going to induce a bias in this beta variable. Okay, that's our source of confounding. If I replace x with the expected value of x given z, then essentially I've thrown away this confounding over here, right? And then these two things are independent by construction. And so replacing the sort of observed x, the x that we actually observe in the data, with the output of a first stage regression, and then regressing x on y, the predicted x on y will give you an unbiased estimate of the truth. So basically, sorry, I said that in a jobbled way, basically just run a regression between z and x, take the predicted values for x instead of the actual observed values for x, regress that on y, and you get an unbiased estimate of the true beta. Okay, that's two-stage least squares. So we can see that again. So here this is the sort of predicted values for x given z. I've chosen like a discrete z because it makes things nice and easy to see. So z just has 4 different values it can take on, and so we just get 4 different predictions essentially. So that's what this prediction happens. And then if you run a regression on this now predicted values, you get an unbiased estimate of the truth. So this yellow line over here is the two-stage least squares. And so this is instrumental variable methods. We have instruments, I said, and so there's a sort of question is like, are we done? Right. Now, clearly, I wouldn't be telling you this if we were. This is this is a very standard method in causal inference. So what is the problem here? Well, the problem is that unless we have a lot of antibiotics and we run a lot of experiments, we're going to have less instruments than we have treatments, right? And so this matrix over here for this estimator, so this is the estimator that we need to get this estimator of the treatment effect. This matrix over here is not going to be invertible, okay? And so kind of more. So that's the sort of technical problem. So we can solve that technical problem by being kind of machine learning people and just replacing when we hit a technical and like a non-invertible matrix, we just replace the inverse with a pseudo inverse, so that we can numerically solve that problem if you want. But then the causal question that remains is, well, what happens when you estimate when you run this estimator with a pseudo inverse instead of the real estimator? The real inverse. So, okay, so we can do, so the problem is this problem is underspecified, right? So there are infinitely many possible betas that are consistent with the data that we've observed so far. And by taking the pseudo inverse, what we're doing is we're picking the minimum norm solution, the minimum L2 norm solution of this problem, okay? We can show, and we showed in our ICML paper, that this estimator is asymptotically normal, but what it does is it converges not to beta, but a projected version of beta, and it's a projection onto a subspace that I'll explain in the next slide. So we don't get the true beta, we get a projected beta. And this projected beta is kind of easiest to see by example, so that's what I'm going to show now. So again, take this very simple case, and for visualization purposes, just imagine we had 2 bacteria. So these are 2 treatment variables and 1 antibiotic, because that just makes it easy to visualize. So our treatment effect over here, this beta would be effect. This is the sort of the first coordinate would be the effect of the first bacteria on intelligence. The second coordinate would be the second bacteria on intelligence, and that's the thing we're trying to estimate. So that's this green vector is the vector that we're trying to estimate. And then alpha over here is the antibiotics effect on the bacteria, right? So this is how much the antibiotic 1 is the antibiotic affects bacteria 1 and bacteria 2 respectively, and that's what defines alpha, right? And the projection that we do is we project onto the space spanned by alpha, right? So if we sort of consider different values of the antibiotic, think of this as like different dosages of the antibiotic, what is the sort of line that we sketch out? And that's the subspace that we're going to span, right? And by taking the pseudo inverse, what we do is when we run this estimator, we get the beta that we're going to get out is this original beta projected down onto this space that's spanned by the instrument, right? And as a result, for different alphas, we're going to get different estimates of beta, right? So sometimes we're going to do a horrible job of estimating beta if our antibiotics have the wrong effect, and sometimes we'll get really, really lucky, and we'll estimate the true effect, right? So in principle, if you got exceptionally lucky and our antibiotics just happen to line up with the treatment effects in the right way, you would estimate the true treatment effect, and you could do that with a single antibiotic, no matter how many instruments no matter how many bacteria you were targeting. But you would have to get unbelievably lucky, and sort of measure zero lucky, in order for that to happen. And so, the thing to take away from this is that it's like, in principle, it's possible to solve this problem with fewer experiments than there are treatment variables. But we need to know but we'll never know whether we've solved this problem or not without some kind of criteria that tells us whether we've actually estimated the true treatment effect or whether we've just estimated an approximation. Okay, so that becomes the research question, and the thing we're going to leverage is the fact that projections can only make vectors, the norm of the vectors, shorter, right? So whatever our projected beta is, the norm associated with it is going to be shorter than the norm of the true beta. And then secondly, each time we run an experiment, the space that we're projecting onto is going to get weakly larger, right? As long as we don't have, as long as we're running linearly independent experiments, we're going to sort of span a larger and larger subspace. And so the projection is the projected norm is going to get longer and longer and longer as we run experiments, right? And so we can see this visually in this 3D case. So now we're looking at a 3D beta. So we've got 3 bacteria affecting intelligence. And if we run just one experiment, I project down onto the space span by that first instrument. For simplicity, we've just put this along an axis aligned, but it doesn't have to be axis aligned in this case. I run a second experiment, then now I span this whole plane, and so my projection gets a little bit longer. You can see that norm of that vector is longer than the norm of this vector, and sort of weakly gets longer. And if we were to run a third experiment, we would identify beta exactly, because then we have the right number of instruments. And so this suggests kind of a natural approach that you might take. Well, you keep running experiments and you keep watching this projected norm. Each time you run an experiment, you get this projected norm. But then the problem you face is, like, if I get to this point, am I kind of done? Is this the norm of beta, or do I have to keep running for a lot longer? And will I ultimately get up to a norm that looks like that? That's then the second question. And so if we knew the norm of beta, we'd know exactly when to stop, right? We'd know when we were done with experimentation. And it turns out that if you're prepared to make some more distributional assumptions, Dominik Janzik has a paper in 2019 which gives an estimator for the norm of beta under sort of like an additive mixing assumptions, and so that then gives you an stopping criterion. Otherwise, you have to stop basically when you think that you have converged, and that's obviously a more hazy concept. right? And so you can put this together into an algorithm, and so that says, and so the algorithm says, assume you have some similarity metric over the antibiotics. Let's explore it a little bit more efficiently. So we can always kind of select the most dissimilar antibiotic. And then in each round, run another experiment and update the estimate of beta, and then stop when your projected beta is approximately the same as the true beta, right? And one thing I forgot to say is, okay, so this is an algorithm that we can run, which is nice. But the one concern you might have is what happens if I run out of money when I'm running these experiments before I've kind of hit that stopping criterion? Is the entire set of experiments wasted, right? Because I'm only getting these projected betas. And for example, like if I was in this situation over here, where the true beta is the blue line, the estimated beta is the orange line, maybe that's not so bad. I'm getting the sign of all the betas right? I'm sort of seeing that this, remember the betas correspond to the sort of size, the causal effect, and so I'm getting the sign right. They kind of don't know whether I'm in this situation or I'm in this situation because they both have the same length norm. And this one's a pretty horrible estimate. I've got one of the signs wrong, as I'm very far away from the truth. And I can't tell these two things apart from data alone. The one thing that you can do is you can check whether any of the standard bases, so this is the x direction or the y direction would be the two standard bases in this plot, whether those get projected onto themselves. And if they do, then you've identified that coordinate of beta, right? So if the standard bases are projected onto themselves, it tells you that you've at least identified that coordinate of beta if you haven't identified the whole of beta. So that's kind of nice. Okay, so we ran this, and what we find is it converges to the true beta kind of quickly. So this is sort of mean squared error between the true beta and the estimated beta, and it converges in far fewer rounds of experimentation than you would need to exhaust if you were sort of exhaustively running these experiments. And you can see this convergence also in the norm of beta, which is kind of what the thing that we get to observe in practice, right? So this is the error bars over multiple random seeds, where over a number of rounds we see the norm of beta converging to the norm of the projected beta converging to the norm of the true beta as we identify the true effect, which is what we want. Okay, so summary from that section, clearly we want to extend this also to the nonlinear setting. I think that opens up a lot of exciting things, which I'm happy to talk about some more. It's a much more challenging setting to think about because now you have to think about like the whole set of functions that are consistent with these constraints that we have, and I think you're going to need some constraints on that function space because otherwise this problem is hopeless. And then I think it's also worth asking whether there's different assumptions that can we weaken the assumptions that we need to estimate this norm of beta? The Yanzik paper is nice, but it's it's an estimate. It's not clear that it's the best estimator of this norm of beta. Maybe weaker assumptions will make that more practical. I'm going to pause here for a second just to see whether there's any questions on that section, because now we're going to switch gears to representation learning.
There is a question in the chat box. I don't know if you can see it. I can read it out if you.
I've got it. Sorry, I'm very bad at monitoring the chat while I'm talking. The number of experiments depend on the number of bacteria. How do you get the number of bacteria from a high-dimensional observation? How do you know the dimension of X? Right. So that we assume is known. So that we assume you can actually go in and you can take sample of the population from your patient of interest stomach. You measure all the different kinds of bacteria, and we assume that you have access to that. It's an interesting question if you don't, but for the purpose of this paper, we just assume we knew that. Yes, question from Vahid.
Hi, Jason. Thanks for the typo now.
Hi.
I wanted to know that do we here assume that the treatments are affecting the outcome independently, even in the linear setting, or not?
That's a great question. So in the way I've drawn the graphical models, it is assuming linearity. It's not required. But I need to think. The one thing
So I
that's
would
subtle
imagine,
around that is like mediators in that setting get kind of subtle.
yeah, I see. Because if you just want to run a linear regression, if there is like dependence between like different variables, different input variables, then what the coefficient that you get is not going to be necessarily an unbiased estimator of the true coefficient, right?
Yeah. You're exactly right. Yeah, I mean, thinking about the relationships among the treatment variables is something we haven't done carefully. And actually, the other paper that I cited in this thing is from Jonas Peters and someone, I can't remember who else. They've got a nice work that came out exactly at the same time as us, and they were a little bit more careful about the relationships between the variables. They had to assume sparsity, so they, at a very high level, we looking at the two norm, they're looking at the one norm. We get an explicit stopping criterion. There's zero norm, which they approximate with the one norm. But they think more carefully about the relationship between the variables in the instrumental variable setting. Okay. I mean, I think sort of my main takeaway, I think, from this is I think this is a really interesting setting. This like setting where you've got this high dimensional system of interest. We can tick it around with experiments, but we can't experiment on everything. And I think that's a very that's a sort of very realistic setting that we're in, that we just don't have good methods for. And I think these two approaches are kind of like the first stab at that, but I think there's going to be a lot of development in this space.
Okay, I see. Thanks.
Cool. Okay. All right. I'm going to keep going because I realize I only have 20 minutes left of time. So let me let me speed up. Okay, so switching gears completely to causal representation learning. And this is a question of like, how do I deal with unstructured data? And I'm using as an example an image that came from Recursion. This is actually part of the reason I'm at Recursion, is I think the data is ideal for thinking about this. So as a sort of problem to have in mind of the kind of thing we'd like to be able to solve, is we get images that look like this, which are, this is a cellular microscopy image, which is, you can think of just as a photograph of a cell under a microscope that's been stained into particular ways to show different parts of the cell. So the images look pretty. The staining is not actually important from a machine learning perspective. You can get away without stained cells, so-called bright field imaging. And then we can perturb these cells in different ways. So these are siRNA knockouts or knockdowns, and so you with different siRNA perturbations, you see different images. And we're interested in sort of these kinds of like, what is the causal effect of these perturbations on these cells? And similarly, you might be interested in questions like, what is the relationship between cells in the state and some health outcome of interest? These are the kinds of questions downstream we'd like to be able to answer. And now clearly we don't care about the causal effect on the pixels. That is not meaningful. And the way we tend to think about this in causal representation learning is that there are a set of latent variables that sit underneath the underlying the pixels themselves, right? So you can think of these latent variables as corresponding to some description of the underlying cell state, and then our perturbations are affecting that cell state. Now, there are a lot of complexity involved in these images. So for simplicity and to make it easy to talk about, we're going to use the most trivially simple images for most of the remainder of this talk. And that's where our latent variables. Question from the chat. Yes, these are images from a cell painting assay. That's correct. But the problem setup is not specific to cell painting. It's kind of any latent variables that spit out some pixel level data, right? So in particular, here is like a trivially simple version of this problem where we've got a set of, I don't know, about 15 latent variables that generate these images, where the latent variables are just the XY coordinates of each of these 5 balls. So that would be 10 latent variables, and then maybe the hue of each ball to give us color information. So that gets us up to 15 numbers. And then the question we're interested in is, when can I recover these latent variables with identifiability guarantees? So when can I sort of provably design a method that's going to recover these latent variables, right? And the first work that I did in the space was work that I did with Kaji Kahuja and Yoshua Benjo when I started my postdoc at Mila. And here we were interested I'm sorry, but before we get to that, I just want to give a warm-up, which is sort of why is identifying these latent variables hard, right? And what we'll do is we'll look at perhaps the kind of canonical machine learning dimensionality reduction technique, which is an autoencoder today. And so we take our image like this. Now, as a thought experiment, let's imagine we start with just the perfect encoder, the thing we'd like to have, which is some function G inverse, and I'm calling it G inverse for pedagogical reasons, because I'm gonna think of it as like inverting the data generating process. So I have my encoder that maps from pixel level data into latent space, right? And this latent space is going to be the XY coordinates and the color for each of the balls in this image. And then it's an autoencoder, so I also have a decoder that maps from this latent space back to the image. And the only constraint that I'm placing on this model is that it has to perfectly reconstruct the data, right? So it has to sort of be the identity function, okay? And so that's this would be great if we found it. This is the thing that we want. But the challenge that we face is if I just apply any invertible transformation A, so if I just do some arbitrary nonlinear warping of this latent space, and then do the inverse of that arbitrary nonlinear warping of this latent space before applying my decoder, then because A inverse A is just the identity function, I haven't changed anything with respect to my reconstruction objective, right? And so now I've defined a new encoder and I've defined a new decoder, which still satisfy the only requirement that I placed on this model, and yet is sort of arbitrarily non-linearly entangled, right? And I can do this for any A, right? So for any nonlinear transformation that's invertible of the latent space, I get another encoder-decoder pair. And so I have kind of uncountably infinitely many solutions to this problem, and only one of them is the one that I actually want, right? And so that's the identification problem. And this isn't just a problem with autoencoders. If you prepare to assume that your latent variables are independent, then the latents are still not identifiable from IID data. So this is an old classic result from the nonlinear ICA literature. And so this then raises the question, well, okay, so if it's not independence, kind of what do we need? Now, this has become a really, really active area of research. The slide is totally out of date. And rather than updating it, we've just made, Danya Shrida and I have made a nice tutorial, well, nice in our biased opinion, where we've gone through a lot of these methods in detail. So if you're interested, this is the tutorial was originally given at UAI, and we gave it recently at Mila where we recorded it. And so there's a bit.ly link, or I'll share these slides as well, where there's an explicit link to this tutorial. So I'm not going to cover all of this. I'm just going to cover, give the very biased view, which is cover my own work in this space. And so we're going to cover how knowledge of mechanisms in this space constrains latent variables, and then how sparsity helps constrain latent variables. Okay, but generally speaking, what we're going to be doing in order to make this problem identifiable is placing some distributional constraints on the latent variables, or alternatively some constraints on the functional form of the mapping, the true ground truth mapping from latents to observations, or some combination of the two, and that's going to let us identify latent variables. Okay. All right, so the question we're interested in properties for mechanisms was how do the sort of mechanisms that govern a system's evolution constrain representations, right? And as a toy problem, imagine these balls bouncing in the background as kind of the toy problem to have in mind. Their dynamics are controlled by the physics of the system, and we're interested in how kind of knowledge of those physics of the system would constrain representations we can learn. So abstractly, we have latent variables that evolve according to some mechanisms. These would just be like the transition functions in a state space model, and we're going to assume some knowledge of these mechanisms, and then what we get to observe are the pixel level representations of the state, right? So the state's evolving according to some mechanisms, and we're observing the pixel level representations, and then we're trying to learn an encoder that maps from pixel level data back to these latent variables. And we're going to assume we have access, we're going to assume that the rendering engine, or the sort of mapping from latent to observations, stays the same across observations. And then we're going to make different assumptions on the mechanisms and when they're modified. So we're going to place some constraints there. Now, to see how this is going to be useful information, let's do the same exercise we did before. So again, start with a perfect encoder that maps to latent space. And now for kind of to ask the extreme version of the question, let's just say we know the mechanism, that the function that takes us from time t to time t plus 1. So if we knew it, we could just apply it to latent space. And what we'll get out is what we would have gotten if we had encoded this frame, right? Because we know the encoder, we know the mechanism, so we get the sort of latence associated with the next frame, right? And then we can decode these and get, the next frame's pixel level representation. And so we can ask for the same kind of reconstruction objective that we had before, except we're going to stick the mechanism in the middle, and we're going to encode the frame at time t and decode the frame at time t plus 1. Okay. And then you can ask, well, does that solve the problem of those arbitrary nonlinear warpings, right? So if I applied that arbitrary nonlinear warping that I had before, you're going to see that we no longer have equality. And there's sort of two ways to see it. One, graphically, the arrows no longer line up, right? Our orange points are not the same as where the mechanism is pointing us to. And mathematically, it's probably easier to see. The problem is that arbitrary nonlinear mapping is not going to commute with this additive offset mechanism, right? So if I can't just do the cancellation that I had before of A inverse A being the identity function. So because A doesn't commute with M. What does commute with M? Well, in machine learning, we often call sort of transformations that commute with some function equivariances of that function. And so those are going to be our source of non-identifiability. So for example, a shift in this mechanism is going to be an equivariance of this mechanism. And so we're going to at best be identified up to those shifts, right? So the sort of the best thing we're going to hope for is that we're going to identify these latent variables up to equivariances of this mechanism. And the thing that we showed in our paper at ICLR last year was that when the mechanism is known, those are the only sources of non-identifiability. So you identified up to the equivariances of the mechanisms. We also had analogous results for unknown mechanisms. So you just have a hypothesis class of the mechanisms rather than known exactly, and also for the stochastic case. Now, I've told you that we're identified up to the set of functions that commute with the mechanism, right? But what functions are in that set? Well, I'll give you some examples here. So if I don't know the mechanism exactly, but I know that it's linear, and I have a number of different offsets, right? Then what we can show is that you identify the latent variables up to a linear transformation, right? So if this is the true latent variables, the thing I'll estimate might be some linear map of the original latent variables. So we've dealt with the nonlinear arbitrary things, but we still have some linear transformation. In disentanglement, we wouldn't call this disentangled because there's still, each coordinate of our estimated Z might be some linear combination of the original coordinates. So they're not disentangled, they're still rotated in some bases. And if you want actual disentanglement, where you sort of actually have a correspondence between each coordinate that you estimate and the true coordinates, then we need to impose additional structure. And that's what we did in this follow-up paper, where we were thinking about cases where we had sparse perturbations. And the sort of toy problem that we had in mind was we were in a setting where we can take actions that perturb the system in a sparse way. So, I have a game controller that is shifting around these different objects, and I see the sort of dense pixel level representation that outputs. And you can see this a little differently here. So the actions I take might shift, say, the color or the position of the objects, and this can have dense pixel level effects. But we know that we're prepared to assume that what happens in the latent space is sparse. And what this allows you to do, what the sparsity allows you to do, is it allows you to reject the sort of rotations that we had before from the linear identification, right? Because if I've now got pairs of frames that I know have changed sparsely, I have to choose a coordinate system that allows me to only have this sort of sparse change, right? So if I had chosen a sort of rotated coordinate system, which would be illustrated on over here. Sorry, there's a typo here. That should be Z1 and that should be Z2. Then I'm not representing this change sparsely, right? I have to change two variables in order to make this change. Whereas if I'd chosen a coordinate system that is axis aligned with the original coordinate system, then I only have to make one change, right? And so this ends up being a very useful, a useful sort of sparsity ends up being really useful, and you can show that if you've got one sparse perturbation, then you identify the true latents up to permutation and scaling, which is kind of the strongest result that you can hope for. So you never know the sort of original permutation of the variables, and you can't pin down the scale, but otherwise you get the variables exactly. If you've got K sparsity, you get up to linearly entangled blocks, and when the blocks overlap appropriately, you get permutations and scaling. Okay. Now I realize I'm basically out of time, so I'm gonna have to skip the last section. But I think I want to make these comments, which is that the community, the causal representation learning community, has made massive progress in the last couple of years in understanding the conditions under which disentanglement is possible. But I think there's still a big gap between the gap between the distributional assumptions that we make for like identifiability proofs and the distributional assumptions that we're prepared to make in practice has mostly closed, right? There are methods that make assumptions that are sort of almost what I'd be satisfied with in the cell painting assay that I'm motivated with. In particular, there are a couple of methods out there now that work in the so-called interventional setting, which is where all I've done is I've just intervened on a single latent variable and seen the resulting observations. And that maps very nicely to the biology setting where I do like a gene knockout, which is just targeting a single gene, and then I get images. But evaluation of these methods is still limited to synthetic tasks where we know ground truth. And on real data, this is still a real problem. And so I had an intern, whose work I'm not going to get to talk about now because I've run out of time, where we've spent the summer trying to apply these in Recursion's data. And really the problem becomes about evaluation, because these methods, even though they give guarantees of recovery, in principle, in finite samples, you don't know whether they've succeeded or not. You don't know whether your distributional assumptions match the real data's distribution, and so you get out an estimate, and then evaluating whether that estimate is good or not is something that's really hard. And so, unfortunately, I'm not going to get to talk about the matching work that we did, or talk about the fun slides that I made up for this. But I'm gonna finish it. I'm gonna finish at this point, and happy to take questions at that point.
Wonderful, thank you. Let's everyone thank our speaker for a wonderful talk. And if there are more questions, hold on, let me just give me a second. If there are more questions, please, you can raise your hand or type in the chat box. I think we still have a couple of minutes, maybe. I know our speaker answered some of the questions during the talk, but I think if you have more questions, you can ask now. And in the meantime, I have a quick questions about the first part of your talk. A lot of the intuition that you used to build up your models were based on the linear assumption. And at the beginning, you mentioned that you've also done work on the non-linear like removing the linear assumption. I'm having trouble finding how the intuition translates to the nonlinear models.
Good. Okay. So I can say how the relationship between the linear and the nonlinear setting when you're identified is relatively straightforward. So when you don't have this problem of being underspecified, the correspondence between, so in the two-stage least squares was the method that I showed, which was where you just sort of you rather than using the actual x that you observed, we use the estimated x from a first stage regression that essentially just kind of projects out the confounders effect on the treatment variables. And so then you can use that as though it was the real x that is unconfounded, and use that in a second stage regression. That doesn't work in the nonlinear setting, but what you need to do is you can do a similar thing by just fitting by doing sort of density estimation of the first stage, and then taking samples from that density estimator and fitting a second stage problem. So you kind of don't have linearity of expectation. So you can't just do this in sort of two explicit regressions, but you can still push through these methods to the nonlinear case, and there's been a lot of nice work in that space. As far as the sort of like underspecified case in the nonlinear setting, I think the nicest way of thinking about this is sort of from a kernel's perspective, or at least basis function expansion perspective. The challenges of nonlinearity are that you can now allow for interactions between variables and interactions between things that only show up in pairwise perturbations that don't show up in individual perturbations, right? So if everything's linear, if I run an experiment, I perturb everything, and there's no sort of interaction effects. It's all just everything's nice and linear, and I can see kind of how I pushed my system around. If I'm allowing arbitrary nonlinearity, I might need to run like multiple antibiotics at the same time in order to see the effects, to see some treatment effects that it would just like not be visible if I ran the sort of three antibiotics separately. So that becomes much more challenging. You also potentially need a lot more experiments because you kind of have to sketch out. If you take like a basis function expansion view of this, so you think like maybe I have to get like the linear term and the quadratic term and the cubic term and so forth. It just blows up the space sort of dimensionality of the function that you're trying to estimate. So rather than just the dimensionality coming from the number of bacteria that you're interested in, it's also from the sort of nonlinearity of the function as well. And so there's sort of these two ways that this gets even more complicated. We're working on it. We haven't figured it out properly yet.
Thanks. Really interesting. Maybe one thing that could simplify a little bit is that probably in many cases we're not interested in any arbitrary nonlinear mechanism, like maybe something like monotonic or saturates that could sort of like we can go from linear to something that is a little bit more relaxed but not totally arbitrary.
Yeah, so that's something I really want to do. We haven't done that, but I would love to work on a particular problem with this set of domains and this set of techniques where with a particular problem in mind, you might say, okay, well, I don't care about any arbitrary nonlinearity, any monotone function that sort of saturates in the right way is fine for this setting. I don't expect my treatment effect to be sort of some weird sinusoidal crazy thing. Yeah. And so, yeah, that's definitely something we should do.
There are also a couple of raised hands. Matthew, maybe you can go first. You can unmute yourself, and if you feel like it, you can turn your camera on as well.
Yeah. Sorry, I unfortunately don't have a camera on this laptop. But sorry about that. I just wanted to ask really quickly. Fascinating presentation. I'm curious. You mentioned in the second part, you were talking about how we have the equivariant functions, and if we know that our mechanism is linear, then we know we can recover up to sort of the linear transformations in the latent space. I'm wondering, do we have any information on, in general, if we can do that, or is only the linear case been studied, or are there any other rules for if information we know about the mechanism can provide information about the latent space kind of recovery?
The short answer is no. So this is something that like we've actually been wanting to do. We just haven't kind of gotten around to it. Is thinking about more general functions in general classes of mechanisms beyond linearity. My conjecture is that if I know it's nonlinear, I think you actually get stronger identifiability results. If I know the right function class and I know, so like if I say, for example, know it's quadratic and not linear, I think that actually might rule out the rotation. But I'm not aware of anyone who's actually studied this carefully. And I've looked into, there are results, like particularly for polynomials, there's sort of like results for kind of what polynomials commute with what polynomials, and there are very few that commute with each other. It's I can't remember if it was Cheryshev or Hermite, but one of those classes of polynomials do commute, but they're kind of like the only ones that commute. And so particular class of non-linearities might give you stronger results, but then you need to know that mechanism, right? So it's sort of like you need to find yourself in a setting where you know the mechanism that gives you these strong identifiability results. If you're trying to learn these both jointly, then the more flexible you make the mechanism, the weaker your identifiability results, because now you are allowing for more nonlinear entanglement in some sense. I don't know if that makes sense.
Thank you. That's super helpful. Appreciate it.
Thank you. There's another raised hands. Maybe you can turn on your camera and ask your questions.
Hello. Hi, I am Mushfiq. So I have a more general question. So in defined causality problems, for example, like causal identification and everything, we need the causal graph, right? So when the variables are suppose low dimensional binary, we can run define PC algorithm or FCI algorithm to get the causal structure. But suppose in some data set we have both types of data, for example, high dimensional variable like images, and also maybe binary variable, something like that. So in those cases, we cannot run PC algorithm or those types of causal discovery algorithm. So I was wondering, can we use causal representational learning to model this causal relation between different types of variables, like between image and binary variables and something like that? What do you think about this?
Yeah, so I mean, I think that's the kind of key motivating goal of the causal representation learning community. Like I think the dream that we all have would be, can we find algorithms and assumptions that we're satisfied with, that lets us recover latent variables and identify the graphs in the latent space at the same time. It's very nicely articulated in that position paper from Bernard Chokov and Joshua and their respective students, which towards causal representation learning. And I think that's the view that they articulate and they push. I think the main challenge is just, at least from my perspective, having tried to, basically the reason I went to recursion and to valence was because I thought that they have the kind of ideal data for this. And I've been very curious about can we make these methods practical? And I think testability is a real issue, and we need these methods are not testable, and they're not testable. Causal inference always has a testability problem, right? If I'm just doing classical causal inference, with from observational data, I still have a testability. I don't know what's happening on the interventional distribution, but I could at least in principle run a randomized controlled trial to test my estimate, right? So if I give you an estimate from observational data, I can then go and run a randomized controlled trial and check that it's right. We can't do that with causal representation learning. And so I think a big gap that is missing is, how do I make these methods trustworthy, so that the thing that I estimate, I know that is true. But yes, I think your question is right. Like I think that's very much in the spirit of what we're trying to do here. But I think we need ways to make these methods, the output trustworthy.
Yeah, I think like the challenges with real data is still existing. Like, how can you place the correctness? Yeah, I think I got my answer. Thanks.
Cool. I see there's a question in chat from Vahid saying, this might sound a more philosophical question. I love philosophical questions, so let's go. The inductive biases, such as sparsity of the interventions, are essentially coming from the human intuition, which is built based on data interventions we have observed in the past. What if we give the current models without these inductive biases the observational and interventional data we have and let them find the best representation? I think my question is more about the practical implications of causal representation learning will result in more interpretable models or models that understand the world better. Yeah, this is a great question. It's sort of my slides kind of allude to this question in this concluding slide. So I'll use the last point as kind of a way of answering this question. When we're running a representation learning algorithm, we're essentially choosing a coordinate system on which we can then learn the mechanisms, right? So we're saying, this is the representation of these are the sort of objects in the scene, and then we can learn mechanisms on top of those. And if I change the basis, I can change the mechanisms, right? And this is sort of a standard thing that's well known in physics. The choice of basis can have a huge effect on the mechanisms you learn. So my favorite example of this is the geocentric and heliocentric worldview. So I've got these pictures over here. On the right we have a geocentric worldview, which has the Earth at the center of the solar system, and on the left we have a heliocentric worldview, which has the sun at the center of the solar system. So basically just a change of basis, right? Both admit perfect prediction of the movements of planets. If you add enough epicycles in the geocentric worldview, you can still perfectly predict the movement of the planets. This is not a failure of prediction. But we prefer the explanation on the left because it admits this, in part because it admits this like really nice, simple functional form that explains all the motion of the planets, right? So if you take the Newtonian view, because that's as far as I got with physics, we can just describe the motion of the planets in terms of their distances and their masses, right? And that comes about by choosing the right coordinate system. There is clearly, having said that, like there is clearly a strong bias in our sciences. We have studied systems that we are able to understand, and we're able to understand things that are kind of nice and easily representable in our in language that we can talk about, right? We've made far more progress in physics than we have in biology, I think in part because physics admits these really concise, beautiful explanations in a way that like complex systems like biology or economics just don't, right? And so it's far less clear that like simplicity is a universal property or just a property of physics, right? And so we have to sort of be careful not to overfit to simplicity as being the right sort of philosophical mode of thinking about these things because it's worked so well in one science, and it's not clear that it generalizes to other sciences. So yeah, it's a great question. I don't have a good answer.
Very good. Thank you. I think it's almost 15 minutes past the time you allocated, so we don't want to take too much of your time. Let's everyone thank the speaker one more time. You can do it either this way or with the icon. Thank you. Really appreciate it. The talk is recorded and will be posted, so you can share it with your friends who missed the talk. Okay.
Awesome. Thank you so much. And thanks so much for having me. This was really fun.